I want to talk a little bit about decision making and how that made such a difference this year, how we make decisions and how it informed how we did things, and a little bit about teamwork, a little bit about leadership, and how our staff helped out and so on. Talking a little bit about the various people that helped us and supported us. So we're going to touch on a lot of points, and certainly looking at the learning from the last year and how we'll be going forward. So. Um, it's going to be, I think, a fairly positive talk because I'm a generally positive person, I think. And there's a lot of learning, and certainly Paul Reed on the first night was talking about how we can move on and change things, and how Sláintecare has been, partially delivered through this, and so on. And there are opportunities in all crises to learn and to grow. And I suppose that's the way we try to look at things. We try to be positive. We try to grow all the time and improve things, and learn from things that could be done better. So it would be fair, I suppose, at the start of this to acknowledge, while I can talk about the positive aspects, to acknowledge that there are also negative aspects to COVID. And without laboring the point, there are many families who have lost loved ones, and many people who have serious disease and who have been left with morbidity following this disease. And just to acknowledge that, and we're still caring for some of these people, and it's important we acknowledge it, and we don't dismiss that while we can talk about the positives. So just to acknowledge that from the start, and I'll move on. The operations manager, what is an operations manager? Well, I looked up definitions for this because I do have a job description like everybody else, and it covers a very, very broad range of things. And all of the definitions of operations manager do cover a broad range. They cover the likes of financial management and accountability, things like coordination and supervision, things like compliance with standards and that type of thing, productivity and output, and being responsible for the flow through the system and the output that we produce, which is a service in this case, communication in the organization, strategic development and planning, project delivery, and that's a lot of stuff for one person to do. So I suppose in a smaller organization, the operations manager may be all those things and may do all those things. In a larger organization such as CUH, with over 4,000 people working here and huge throughput of activity, you tend to have other people doing a lot of this work, and you're responsible for it. And so you're keeping an eye on the outputs and the data around that. So we have a services manager who minds the compliance bit and the health and safety and all that type of stuff. We have a HR manager, we have a finance manager, and all those people in some way report to me. We have a bed manager who's part of the nursing complement, reports to the director of nursing. And at any given time, we have a whole series of data and metrics that tell us the temperature of the hospital as it stands. And it tells us where we're doing well and where things are probably maybe not doing so well, and where we have to put our efforts to improve things. And therefore, your accountability is all wrapped up in that, knowing what the current situation is. It's also wrapped up in each of those people being really, really good at their jobs. And we're very fortunate to have that in CUH. We have extraordinary people working here. And, for my part, I try not to micromanage people. I try to choose people for those roles that really do a great job and don't need a lot of support, but we keep in contact, and we keep regular meetings and so on. And that's how it works here. I suppose one of the definitions of operations manager said that you're the glue that holds the organization together, and that's quite a quite a title to have, and quite a responsibility. But I don't know if I see myself as that, but certainly I'm a touch point for a lot of those services. And when there's a complicated issue to deal with or whatever, I'll often be involved in trying to sort that out. And it's quite a varied role. My day changes. Every day is different, really. So who am I then, and where did I come from? Well, Brendan O'Reilly was a medical scientist, or originally a medical laboratory technician, and maybe that's one of the reasons why the Biomedical Society spotted me or picked me out. But I started out in 1989, before a number of people here, I'm sure, were born, as a graduate of the certificate course in medical laboratory science. And I've worked in labs for most of my life, for 27 years. And I went through various promotional grades, senior grade and surveillance scientist and so on. I became chief of the micro lab for a few years, and I was also laboratory manager over all of pathology. I spent most of my career in CUH actually, and so I have a strong lab background. I still do a little bit of lab work assessing labs for accreditation to ISO 15189 on behalf of INAB, the Irish National Accreditation Board. And in my private life, I'm an emergency medical technician, which is you see these on the intermediate care vehicles. So this is a person who can deal with a medical emergency in the community, and I do deal with those. And so it's been, the reason I mention it, I suppose, is because it's been of great use to me in my job to have had that medical background and to feel as though I'm a person, a clinical person who manages, as opposed to simply a manager. And so that's important to me. And I believe it gives me currency, and it helps me when I'm dealing with clinical people, and it helps me understand where they're coming from, and it gives me credibility and so on. So it's very important to me. It also helps me distinguish when we're talking about decision making, when a decision needs to happen right now, when it's a critical decision, when there's life or limb at stake, or when we can spend a little time gathering data and figuring out the best course. And that's important to recognize those things. I mean, just by background, I'm also married, and I like to play music in my spare time. Look, that's my journey. I suppose I'm 3.5 years in the operations manager role now, and it has changed quite a bit for me in that time. I'm still learning, and always will be learning. I'll be lifelong learning. But I try to be accessible. I try to be supportive of the staff, and not overbearing, and not trying to do their job for them, because they're great people, and try to be as hands off as I can, as long as I know that things are running okay. And that's, I think, the best formula. I was walking down the corridor during the week with one of the clinical directors, and we were chatting about the last year in light of this talk. And I said, well, what did I do for the last year? And he said the same thing. What did I do? What can I point to that was mine and that I owned for the last year? And realistically, I think as you go along in your career and you go into these promotional roles, more and more it becomes about how you've created a space where other people can get things done, as opposed to you specifically doing things, certainly when there's the breadth of what's in this role. And so this isn't an excuse for me not doing stuff for the last year, but this is the way I look back on it, is being part of teams that decided how we would approach certain situations, supporting people who know what they're doing, people, great clinical people who inform us and fill in the gaps that all of us have as leaders and managers, and then making collective decisions. And sometimes there needs to be an ultimate decision that falls to you, but for the most part, we have great people who advise us. And so in the last year, it's very hard for me to pick out things that were all mine. There would be very few, I would say, would have been part of a lot of various projects and so on. And it's been a very rewarding year in a strange way, I suppose. And so just to talk a little bit about decisions and how we make decisions, I mentioned recognizing when something needs a decision urgently, or when something can take a little bit more time. That's really important. I mentioned listening to the experts and listening to the people who really know, and understanding what your own limitations are, and being humble enough to accept input from others and heed their advice. And so we take all that into account when we're making decisions. For all of my decisions in my management career, we'll say, I've tried to keep in mind 3 major stakeholders, which are the patient, the staff, and the organization. And I think if your decisions serve those 3 well, they'll generally be good decisions. So we keep the patients in mind, and we try to build a service around patients. But most importantly, we must protect our staff and make sure that our staff are well looked after, they're safe, and they enjoy doing what they're doing, and they feel part of the organization, they feel part of what we're doing, and that they're listened to, and their input is sought, and they're valued. And the organization, there are principles and values for the organization that we try to uphold, and we try to provide value for money in what we do, and to be compliant, so that we're representing the organization well and trying to align with the principles and the direction of the organization. And so that's really how I try to make decisions and try to keep those things in mind. And maybe the most powerful thing is surrounding yourself with really good people. And I'm very fortunate in CUH to have that. You try to empower your direct reports, the people who report to you, to make their own decisions. And when they have a difficulty, or when they know they've reached a certain point where they need some assistance, they come and look for it. And that's a very healthy working atmosphere, I think. Ultimately, really, integration, interdisciplinary working is really important. In the last year has been a really good example of that. There's been a leveling of the organization. It's been very horizontal. Everybody, I think, of all grades, professional, non-professional, clinical, non-clinical, have had input to how we're doing things. There's a parity of esteem across professions and grades, and it is a much more horizontal organization. I think that's a healthy thing. So I suppose just to go into the year itself and the pandemic, you know, outbreaks and epidemics and so on are not new to the health system. Most people point back to the 1918 flu pandemic. I know John is on here and he's going to speak to you in a short while, and I'm not going to make a fool of myself by talking too much about virology. It was never one of my pet subjects in microbiology, but the way flu reassociates and comes out with different strains every year and so on is a challenge, and you know, hence the annual flu shot and so on. We don't know what's going to happen with this virus. We may end up in the same position where it becomes endemic. That seems to be the common wisdom at the moment. We may end up with annual shots for this. We may end up with escape variants and so on that need a different type of booster or whatever. We just don't know yet. But as I look back over my own career and I think of the early HIV cases we got, there was a time when HIV wasn't a thing, right, for the health service. And coming through in the late 80s, it would have been in the early part of that epidemic, I suppose. And you know, I can remember people asking the question, do we wear double gloves and all this type of stuff? And we've learned so much since then about HIV, and thankfully it's become more of a chronic disease now than it was. We've seen the Ebola outbreaks in West Africa and so on that are never far from the news, and the preparation around that, the training and the donning and doffing weren't terribly strange to us. We've seen multi-resistant tuberculosis, and of course we have the constant norovirus, C. diff, and all these things that are, you know, that the bugs have a way of getting in and spreading, and we try and get better and better, and moving towards a place where we have absolute isolation of patients and where they're protected as best we can. But this is a whole other plane, I suppose, this virus, and it's been a challenging year from that point of view, because this hospital is 42 years old. Parts of it are newer than that, and you know, our infection control practices are so important here, and the training is very solid, and you know, insofar as you can control these things, there's huge effort goes into it. In early March last year, we had one of our first cases, and we had expected this to happen. We were looking at a case coming into ED. We had our plan for which room they'd be taken into, how we'd don all our gear, how that patient, what pathway that patient would take to ICU if they're going to ICU, and so on. We put a lot of thought into this, and we had started seeing patients arriving looking for a swab to be taken, because the St. Mary's Orthopaedic Hospital swabbing centre wasn't set up at that time. And so we were going out to cars. I say we, my colleagues were doing this, going out to cars and taking swabs. One of our earliest cases, of course, happened to already be in the hospital. It was an ICU patient with an undifferentiated pneumonia. And that patient, according to the national guidelines at the time, as we were learning about this disease, wasn't designated for testing. But one of our fantastic infectious disease doctors thought, no, we need to test this patient. We did test. The patient was positive. And the close contacts of that patient who had been nursing that patient were out for 2 weeks. And it took out a lot of our ICU staff isolating in case they got the disease. And that was a huge learning exercise for us. It was a huge learning exercise for the country, and it informed the subsequent testing algorithm for hospitals. And we started to look at surveillance testing of inpatients and looking at patients who might present with atypical disease. And of course, that has evolved throughout the year as to what a pandemic patient looks like, what a COVID patient looks like. And so all our efforts to protect the public and protect the patients, visitors, and that was just an unexpected blip. And this is part of the learning in this, and it was very useful. So our early actions that we started to take, we had a high dependency unit, which is like, which is really these days is an ICU space really, but the high dependency unit in our cardiac renal had not yet been staffed. So that was a plan, as we, you know, a bed is only a bed until you get staff to run it. Okay, so the fifth floor HDU was used for several other purposes, that because in a hospital like this, there's always evolution, there's always services need space, and so we had to clear out that fifth floor HDU and prepare it for additional ICU patients or critical care patients, and it's quite a job finding space for those services, and we did that. We also had, and I think Paul mentioned on Monday, purchasing ventilators in a competitive market where the whole world was looking for these things. So we had purchasing ventilators, we had training staff so they were able to take on a critical care patient. We had things like pumps and this type of stuff that's needed for an ICU patient. We had a lot of stuff to acquire, and there was a huge body of work in that. We also had a huge battle with PPE, like everybody else. Again, it was a global market, and so we started. One of the things that was fairly obvious was a lot of these things were made in China, and China were still reeling from the disease, and so we started to cultivate local options. And we had great relationships with companies locally, which I'll come back to in a few moments. So PPE, we had various sources of PPE coming in, and our director of nursing was exceptional, and our colleagues testing these PPE to make sure they were suitable and safe for staff. And you know, at no point do we leave staff without PPE. I'm not aware that we did, and there was a huge effort on everybody's part, and all those people are to be commended. Like a lot of hospitals, we stopped all elective activity. Now, I use the term elective advisedly. We tend to call activity that's planned, that comes in on a planned basis, elective activity. But of course, we're a cancer center, so if you're planned to come in to have a lesion removed or something, it's not elective in the sense that you don't want to be cancelling that. Yeah, it's scheduled, but it's maybe not so much elective. But we tend to use the collective term elective, and so for a short while we were starting to deal with this, all scheduled activity was really halted, and emergency activity continued. Outpatient activity stopped for a couple of weeks while we figured out what we were going to do, and we started moving towards virtual activity. And the outpatient space, which is adjacent to the emergency department in CUH, the emergency department expanded into that. You can argue that the emergency department is never going to be big enough for what it does. Certainly, it was not big enough at the time to manage two separate streams of patients. So streams of patients with respiratory symptoms or GI symptoms, and streams of patients without those, patients who came in for some other reason. And so our objective all the time was to protect staff, protect patients, protect the public. So we created two separate streams. We used single rooms in the outpatient area and expanded in there so the patients could be kept in separate spaces. We refurbished the entire ED, and all the shared spaces that we had were divided down into single rooms or separated areas, so patients were no longer sharing areas with anybody else. We also had a project on the way at the time, the development of a rapid assessment area that was expedited and finished. For all the years that we lived with the pain, and patients lived with the pain of being on a trolley in a corridor, that stopped. There was no more corridor trolleys for the last year, really, and we're still in that place. So while we have patients on a trolley in a space in the ED who might be delayed getting up to the ward, which does happen, they are no longer on a corridor. The dignity of the patient is protected. They're in a single space, and it's a much better patient experience, and it was one of the significant improvements of the whole process and the whole year. While all this was happening, there was a local convent, St. Catherine's Convent, which hadn't been used as a convent for some time, was refurbished by estates into almost a custom-made, state-of-the-art outpatient. It's not perfect, but it was a lovely space, designed and delivered very, very fast by our wonderful estates people. And so OPD, physical OPD, started up reasonably soon after that, and we had gotten used to starting to do virtual OPD as well. And there's a limit to what you can do virtually with patients. Sometimes you have to see and assess the patient physically. We also designated COVID spaces in A&E. Some of the rooms that we had were designated as COVID rooms. We also redesigned the whole waiting area so patients were separated while they were waiting or while loved ones were waiting for somebody inside. We took over a local coffee area and redesigned that into a waiting area. We also, on the foot of an outbreak in our MSSU, or medical short stay ward, ripped it all out and replaced it with single room spaces again, and we've had no issues up there since in terms of spreading the disease. So there's huge infrastructural change around the acute floor and purchasing equipment to make sure we're able to manage all those patients. Some of the challenges we had, well, testing for the disease was an early challenge. I have to say, Louise gave a good talk on this on Monday, and I wouldn't go over it again. The response from the lab was exceptional. I was very proud to be a member of the senior management team and to have said I came from that lab. And Louise and Dr. Nolan and the team down there just did an exceptional job of delivering on testing for the disease and made it available to us. And of course, the NVRL were just exceptional as well. So the laboratory community were very responsive and very useful for all this, because they gave us answers, which is useful. You know, the configuration of space in the hospital was an issue, because we had to start moving some of those dislocated or discommoded services, and of course we had just vacated our radiotherapy area and moved into the new Glandor Centre, so we had a little bit of space to play with, and that was very useful. What we were not prepared for, I suppose, early on was losing whole teams of people. I mean, the ICU was an example where we lost a lot of ICU staff because they were contacts. And the same thing happened in our cardiology service for a short while. We lost our and they weren't all sick. They were just self-isolating because they were contacts of the patients. And so we learned to step up the PPE early on to protect our teams as well, and to keep them at work and to keep them safe. The oxygen infrastructure was something that we spent a lot of time working on, and making sure that we had enough oxygen to continue with high flow oxygen for patients on non-invasive ventilation and so on. A lot of these patients needed that to keep them out of ICU and to keep their oxygen saturation up. We also brought in additional staff, and I have to say, and I'll acknowledge it in a minute, all the people that helped us, but the HSE were just exceptional in all this. There was no question about the equipment we needed, the space we needed, the support from estates, and the additional staff needed. We were able to take in anybody we needed, and we were very reasonable in all that. We brought in physio people and so on to help us, additional cleaning staff and the whole lot. We reorganized our anesthetics rotas, our medical rotas. Doctors went on to ward-based work instead of team-based. So instead of having a team visiting patients, doctors went to the ward and stayed on a ward, and it was a very good model, actually. And I think it's a model we should use going forward, but we need to work out some of the kinks in it. A lot of departments split shifts so that we didn't have everybody in at the same time. So if there was a contact tracing issue, that we weren't losing the whole team. And as I said earlier on, I didn't micromanage this. Departments did this themselves, and they did it really, really well. Protected us from service disruption. And remote working became a real thing for us, and supporting that, and it's continued on. All the time we were focused on protecting patients, protecting staff. There are probably some fairly draconian visiting measures, and it's not pleasant, and it's hard on staff, it's hard on patients, it's hard on families, but it's all about protecting patients and protecting visitors, and we're doing our best and to do that, and people have been very cooperative with that. And communication was a big part of it. We had daily meetings in the auditorium, where the CEO would give people the current status and hear reports from the various departments, ICU and microbiology and so on and so forth, and share that information across the organization. Line managers then went out and told their staff. So we tried to keep the communication flowing so that people knew what was going on, and the executive leadership, the combination of our clinical directors and our nursing leads, our HSCP and the managers and so on, was powerful throughout the whole thing. And of course, we had a lot of IT. We developed a lot of IT using the likes of iPads and so on for communicating with families, and that was an important part of what we did to keep people connected and that, you know. So I suppose we had to respond to the trends as the waves came and the waves left, and as community cases rose, the activity here dropped. The A&E presentations dropped to about 50% of their normal value, because people probably made decisions not to come in because their injury wasn't severe enough. But some people probably made decisions not to come in because they were worried. And we were worried about that too, because there were people with genuine disease that probably weren't presenting, and there's some evidence to support that now. So our general activity dropped quite a bit. The community services were very, very helpful in taking patients out of the hospital when they didn't need to be in here. They no longer needed level 4 hospital care. And so a lot of patients were taken out appropriately to nursing homes, community homes, and so on. And then, of course, as the community cases started to drop, the activity rose again. But just a word of caution, I mean, the activity, the cases have dropped these last few weeks in Cork, but we're still nursing some really sick patients here in our ICU. The ICU is a lagging trend. It follows behind the community activity, because patients, when they go into ICU, often take quite a while in there to come right, or some of them sadly pass away. And so it was a year where we had decisions to make about when we escalate and when we de-escalate, and decisions to make about how we best try to address and catch up with that scheduled activity, which doesn't go away and is really, really important. And that was a big challenge for us, but we approached it with the same decision-making methodology I mentioned earlier on. And Paul Reid mentioned on Monday that we try to get 70% of our decisions. We expect to get 70% of our decisions right, and 30% may be wrong. And that may well be the case. And at some point, I'm sure we look back and learn what was the right thing and what was the wrong thing. It's very hard to sit back and say, would it have been different had we done something differently? But we made those decisions as we were going along. I suppose during the year, we learned a lot and changed a lot. There was a whole underfloor of an old ward that was disused in the hospital. It was basically a construction site. Our estates people came in and regenerated that into a new ward, which became the COVID ward, actually, the ground floor C ward. We also had some beds that were less used on the first floor, generally used for expansion of pediatric patients during the winter, pediatric presentations. This year, the bronchiolitis cases and so on didn't come as much. So we were able to use that space for medical patients. And there was, again, estate's involvement in making access to that place and dividing the pediatric and the adult population safely. We did a lot of work on our level 5 HDU, bringing in air handling into rooms and so on between waves 2 and 3. And we did work on our protocol and our plans, our triggers, as to when we start to de-escalate and escalate and so on and so forth. A lot of training went on for people to enable them to look after critical care patients. And that was the year really, with the waves of activity coming and going. So I want to mention for a few minutes, we're nearly there now, the people and supports that we had. The staff were just outstanding. We really have great people here. We're very proud of them, and they performed really well. They're tired now, they sure are. It's been a tough year, a year when it's hard and to take time off. You know, people don't want to be let the side down, and that we have encouraged people to do that. We've offered support to people in terms of employee assistance and so on. Some people have taken that up, and it's a healthy thing to do, and some people maybe don't need it, and we encourage people to mind themselves and mind their health. Estates have been absolutely out of this world. They've been really, really good, and still are. The estates budget, to be fair to the HSE, has hugely increased, and there's a huge effort in now to prepare for any subsequent challenges like this, to put in single occupancy bed spaces, to put in ICU spaces, to put in community supports for people both to live at home safely, and if they need additional support with their daily living, to live in community homes and that. There was a lot of money and I put into our labs and supports to do testing, and our labs have been really, really good at supporting us, helping us to make decisions about patient movements and that. there were a lot of companies helped us, private companies, giving us parking spaces nearby, there's a lot of food dropped in for people. There was a lot of companies provided excess PPE stock. Some companies provided digital support, and like our charity, for instance, another huge assistance to us, worked with Dell to provide us with an operational hub. That was a really great project, and we have that now to manage our operations daily going forward. So it was a huge investment from our companies and support from our companies, and we really appreciated it. The media were really good in telling the story and conveying the message to people, and helping us keep the disease under control, and so on. The South West Hospital Group, our overarching governance group, and the HSE, there was no problem that they wouldn't help with. There was no resource that we were left wanting for. We really felt supported, and it was very much appreciated. Our consultants, our doctors, the GPs, just did a fantastic job. We always got to remember the GPs. It's a lonely place to be, and they're really an exceptional group of people. Our nurses, our health and social care professionals really stepped up. I think the support services, the cleaners, the porters, the caterers, security, and so on, all those people who never probably expected to have to don in all of this gear and deal with patients on that basis, all stepped up to it. Our cleaners went into rooms where COVID patients had been and cleaned them. And these people are just really, really great people. They're really valued. And, I do take opportunities to thank them individually as I meet them and so on. And that's really important. The National Ambulance Service was a great colleague throughout all this, and it's one of those organisations, independent of the pandemic, that has developed in the last 5 to 10 years in a huge way, and they've taken on additional roles throughout this pandemic that have been very useful in swabbing and vaccination clinics and so on and so forth, and helping us with our conveyance of patients in and out. The public, of course, have been of great use. UCC also has been very supportive of us. A lot of the space that wasn't being used for educational purposes during the pandemic was given to us for our use, and there was no question about it. They were very, very supportive, and we do appreciate that. And our ED really, I suppose, is our ED and ICU probably saw the brunt of this, and have dealt with it really exceptionally. So I spoke a little bit about the past and previous epidemics and so on, and I spoke a little bit about the present and where we are now. We're still nursing patients. We had 20 patients in today, still, from a height of 164 patients. The future, I suppose, we have to invest in capital infrastructure, and I get a sense that the HSE is behind that. There's a new 48-bed single occupancy room hospital going into Mallow Hospital. There are plans for this site as well, ICU and so on and so forth. And I think there's a bright future for the acute hospital sector and for the community. There's a lot of money going to the community, which is of great value as well. And we try to think of patients as not living in the acute sector or the community sector. Patients live across the continuum of care, and it needs to be built around what they need. Our staffing for the future, there's been a huge investment in the respiratory, the endocrinology, and all of these systems that are affected by this disease, cardiology and so on. And it's great to see that going forward, that there's that will to invest in that. And as I said, we've felt very supported. Elements of Sláintecare, the combination of what we do with the community, have come through this and been developed, and it's really been very beneficial, I think, to patients. And we try to think of the pull system, where you pull the patient back to the best version of themselves that they can be following an episode, as opposed to just treating their immediate needs. And that requires integration. It requires cooperation pre and post hospital, and our every effort to keep patients healthy and out of hospital. You don't want to be in hospital. You want to be independent and keep that independence and not lose that. So we try to build our systems around that. The vaccination, I think, will provide a great foundation for going forward. There's evidence already showing that our community homes and our staff are showing fewer cases. There seems to be an element of that down to the vaccination program, which is reassuring, and I think that's going to hit a fairly high pace, in particularly in April. We'll start to see the fruits of that work, and we may end up with an endemic endemic pockets of disease, but you would hope that that will keep the disease under control by the end of the summer. This is in my final few pieces, I suppose I'd like to talk about the learning from the year, and I've spoken about some of these things. The idea that a supply chain from one country is something we need to be thinking about. We need to cultivate multiple supply chains so we're not exposed in the same way as we might have been earlier on in this. I think the agility, the innovation, the creativity, the reduced bureaucracy in getting things done was hugely useful. We were a very agile, responsive organization throughout this. You'd like to see that continue. And I think we need to design our systems to be more agile like that, and to be able to make decisions and get them done reasonably fast. Remote working will be a part of who we are and what we are going forward. And we need to make sure we can keep the culture the same in the organization and keep people feeling connected to what we do and part of the organization. We need to keep that connection with the community and make sure that what we're doing is not about the acute hospital or about the community. It's about the patient and their journey and supporting them. A really important part of our learning is walking the floor. The director of nursing, myself, and the CEO frequently, most days really, walk down to the ED, and we walk into other parts of the hospital and just talk to the staff and listen to the staff. And it is valued, and I've been told on many occasions it's valued, and it's important. You need to know what it looks like on the floor. I've spent time in the ICU, in the COVID ICU, to see what it's like for those staff and to understand what they're facing, and that's really important. There are core deficits. There are deficits in most services. We all want to develop, and capacity often doesn't meet the full amount of demand. So there are core deficits in diagnostics as well that we will be working away on improving on, and that's part of our strategy going forward. We need to fix those things. Really, we need to mind our staff, protect our staff. Everyone says that they're our most important asset. We really believe that. I don't know if it always comes across that to staff that we feel that, but that certainly is my ethos, and we try to live that every day in CUH. I suppose the one final thing I would say about it all is, during the pandemic, particularly in the early parts, there was a unity of purpose. People talk about mission and vision and values, and sometimes people go a little bit cloudy and misty-eyed when you talk about those things, and they seem very academic and fake. But actually, when you have a direction and when you have a vision and when you're united behind that and you're all aligned with it, everybody rows in. And that is what the experience was like in CUH during this. We were aligned the same direction, wanting to protect our staff, protect our patients, deliver care for the COVID patients and for those patients whose care might have been delayed or interfered with in some way. And that was our purpose for the year. And, I hope we got 70% or more of our decisions right. I'm sure time will tell, but it's been an extraordinary year, and very, very proud to be part of CUH staff during that year. Thank you very much. I'm going to pass back to Roisin now.
Thank you so much for that very insightful thought, Brendan. I suppose as biomedical students, it's great for us to see the different avenues that we can go down, and being the operations manager of the CUH is definitely something I never thought we could do. So it's great to see that, and also to hear a side of the pandemic that we haven't really heard of before. I suppose we heard about the doctors and nurses on the front line, but we never heard about the people trying to reconfigure spaces to make sure as many patients as possible could be treated. So thank you for all your hard work and the hard work of all your colleagues, and thank you for coming to tell us about it all tonight.
Thank you.
So if there's any questions for Brendan, you can pop them into the chat now, and we'll come back to them after Dr. Kenny has spoken. So I'll pass you over to Dr. John Kenny now, who's going to talk to us.
Thanks, Roisin. And hopefully you will see some slides coming up.
Yeah, we can see them.
Yeah. Okay, perfect. Right, well, thank you very much. And thanks very much for the chance to chat with everybody this evening, especially at this hour of a Friday evening. So I'm going to give a reasonably quick overview of the coronavirus sequencing consortium. I'm based in Moore Park site for Teagasc, the Food Research Centre there, and I'm kind of going to talk about the when and the why and the where of the project, how we do it. First off, the who. So unlike a normal normal research project, the intention here is to make the data publicly available, right? So normally the process is you write your grant application and you either generate publications or products or whatever it happens to be. And then you sort of keep that information. In this case, we're making the data publicly available through a website called GizAid. So we do the sequencing, we generate that data, and we make that available as soon as we can. And the idea being that other scientists around the world can access that data easily, and that it's part of a global effort. In terms of the groups involved, we draw from sites all across the country. And our project it's normally focused on kind of hospital patients. Really, that's where a lot of our patients are coming from. You'll see the National Virus Reference Laboratory are part of the consortium. They're also doing kind of sequencing within community samples, and we're expecting to be supporting them more in this as time goes by. So some of the consortium, really it stemmed from Patrick Stapleton, who's a consultant microbiologist in Limerick. He contacted Fiona Crispy, who heads up the sequencing side of things in Moore Park. They worked together in the past, Fiona spoke to Paul Cotter, and Paul leads the overall consortium, and said it'd be a good idea to get a national grouping together, and that led to a lot of very engaged scientists. I'd probably reiterate something that Brendan mentioned there about the unity of purpose. We put together a grant application to Science Foundation Ireland. We were lucky enough to receive the funding. The nature of the funding is a bit unusual as well, in that it didn't cover any staff time, right? So all of these institutions, they were all giving their staff time without any recompense for that. But everybody was fully engaged, and they were really on board for the project. So what are we doing? Well, this is kind of an infographic of the virus. Let me see if I can get a pointer up here. Of the viral genome. So the virus is about 30,000 bases, 30,000 letters in length, and those letters they code for different genes, and these genes code for the proteins that make up the structure of the virus. So let's say the gene that's probably, or the protein that's probably most of interest to a lot of us, is the spike protein. And the reason being that that's the one that most of the vaccines are targeted against. Now, we know mutations happen all the time, right? It's a completely natural component of biology. And it's especially expected when you've got a virus that jumps from an original host, and the original host in this case was probably bats, and then it jumps to a new host, us, to humans. So what is the variant? Well, a variant is just a slight change on this genome. So we see, a base change at this particular position, and that's a new variant. It's a new mutant to the virus. And like I said, it's a completely normal part of the process, and we're going to come back to it again. The spike protein itself, the function of that is that it kind of acts as the key to unlock the cell. So it's the spike protein that binds the receptor on the surface of your cells, and it uses that to gain entry into the host. But because it's on the surface, it's kind of a natural target for the development of vaccines. And why do we do the work? Why are we handling the data the way we're handling it? Well, we're kind of doing it really in accordance with WHO guidance. So we're trying to make the data available as quickly as we can, to make it freely available. Can it do things like help predict disease spread, or can it help evaluate treatments? So for example, if you're looking at variants that have a different structure on the spike protein, will that affect things like how effective the vaccinations are? And how do we do the work? Well, if a patient is going for their diagnostic test, Normally what happens, most people will be familiar with the idea, they get a nasal swab taken, and the healthcare worker will take that swab and they'll put it in a lysis buffer. And really what that does is it breaks apart the virus, it inactivates the virus, and it releases the nucleic acid, the genome of the virus. Then it goes through a purification stage, and really what that means is it just gets rid of fats and proteins, and it cleans it up so it's ready to go into the next part of the pipeline. And the next bit is that PCR test that we hear so much about, where what the biomedical researchers will be doing is they'll be looking to detect that genome in the patient swab sample. So in our case, what we're doing is there's an excess of this material left over after the diagnostic test. So not all of it carries through to the PCR test. And we're using some of that surplus material, and we're taking that forward for sequencing to look at the whole genome of the virus. And we're doing something called the Arctic protocol to do that data generation and the analysis. So that protocol, it kind of stems from previous viral work, previous outbreak work, including things like Ebola, Lassa virus, things like that. And essentially what we do is the virus is the genome's about a 30,000 base genome, and that is technically too long to sequence in one go. So essentially what we do is we split that virus genome into small chunks of about 400 bases, and those are overlapping chunks. And then we sequence those individual chunks, and then we stick them back together computationally afterwards. And when you stick them back together, it gives you the mutational profile of the virus from that swabbed patient. Really, it's essentially like a jigsaw, and it looks a little bit like this. If we have the virus whole genome here, and then we split it up into these small chunks, so we have about 100 of these PCR products or amplicons. You can see they're kind of overlapping there. So we sequence each one of those individually, and then when we piece those all back together, it gives us that image of what the mutation profile is for the virus. So why do we use this protocol? It means that we can share it between different sites, right? The nature of the platform that we do the sequencing on, the instrument, is this Oxford nanopore sequencer, and those are dispersed across multiple labs across the country, which makes it easier. It means the work can be shared. It means we can build up some capacity around the country. You're not dependent on one laboratory at a time. In Teagasc, in Ingenuity Ireland, we have a bigger version of the instrument for sequencing, which means it can be scaled up. The same method is being used across other countries, such as in COG UK, the UK Sequencing Consortium, so it allows you to coordinate with other countries. And this is useful for this pandemic, but it's also relevant, that network is relevant for future use as well. You might wonder why Teagasc got involved. Well, we have a lot of sequencing capabilities on site in Moorpark. And we do a lot of work around the role of microorganisms and their interactions with humans, with animals, with the environment. We look at how microorganisms work in terms of food production and food safety. So it's something, that kind of technology is something we've been working at for a long time, and it's something we're very comfortable with. The sequencing technology itself, so we've made those small chunks of DNA. The sequencing itself, the nanopore process, you essentially have this pore, and if you can imagine DNA being fed through the pore, as it's fed through the pore, that's recorded by the instrumentation itself as a series of squiggles, because as it feeds through, as the DNA feeds through, you get a change in electric potential, change in current. And computationally, the system recognizes that the series of squiggles that are kicked out, it can convert that to the genome sequence. So it's the genome sequence that we're interested in and work with afterwards. And that's kind of an idea what the instruments look like themselves. The MinION, which is about the size of a stapler, the GridION, which is kind of 5 of those packed in together, and there's some extra compute on board to help that work a little bit better. And here's Fiona loading one of those systems in the lab. And how do we run the analysis? Well, we use a kind of a comparison, if you kind of call them sort of phylogeny or genetic trees. So if you look at this kind of an example, we kind of intuitively know that certain dogs are going to be more related to each other, right? So if you have something like a West Highland Terrier or Yorkshire Terrier or something like that, you just feel, you know, that they're going to be more closely related. And if we look at their genomes, they are, we can tell they're more closely related because they cluster, they group together when we look at their genomes on a genetic tree. Whereas other dogs, maybe wolves or Dobermans or whatever, they will be more distantly related from terriers. And we could do the same kind of process with a viral sequence. So if we take viral genomes that are from viruses that have been isolated all around the world, and these could be viruses that were found in Africa or America or Europe or Asia or Oceania, wherever, and then we look at the Irish sequences, and these are the ones that are circled here. So these are sequences from samples that were taken before June the 25th of last year. And what we can see is that these viruses are they're interspersed all the way around this tree. They're not clustered really together in one particular group. And that tells us that we had multiple independent introductions of the virus into Ireland during the first wave of the pandemic. And that graphic was put together from more data by Michael Carr and Gabo Gonzalez in the NPR. We can also look at the variants over time. So if I leave here for a second, we'll come to the website. So if we go to the GISA website, and this is the site that we upload our sequence information to. There's another one of these trees over here on the right hand side, and it's not kind of in that circular format, but that's because it's stretched out along here with the time upon which the samples were collected, right? So if we look at maybe one of these ones here, this was sequencing done by Teagasc in Moore Park. The samples came from the NVRL. The sample was collected from the patient in Louth, and we can see some information there about the collection date. So if I press play on this graphic, over on the right hand side, you're going to see a white bar, and that's going to just move along there through time. And what you're going to see over here is you're going to see the virus starting to appear in Ireland. You're going to see some lines which are tracking where that variant of the virus maybe was detected somewhere else and where it's going to be introduced into Ireland. And we're also going to see some lines where a variant was found at one site in Ireland at a particular time, and then it was found at another site in Ireland at a particular time. And when you see those lines interlinking the two, like we've got here, that's kind of an indication that the virus maybe originated in an introduction in Dublin, and then it's been transferred around the country. So that's on the right hand side, we can see movement of time. And over here we have the virus progressing. And the color scheme there, in this particular case, it's based on county, but you could cut the data to look differently if it was of interest to you. Come back to my slides. So that gives you an idea of kind of the dynamics of it, what we call it. You know, if you look at those variants against time, you can also, I mean, Brendan was talking about, you know, local transmission in hospitals. I know group UCD, I think it was St. Vincent's, they produced a paper where they looked at, they'd sequence variants from within a hospital lab. We've done the same thing for Patrick Stapleton in Limerick, where we take samples from particular locations within the hospital. We can look at those variants, and depending on the sequences, we can help educate local responses on a local infection control in the hospital and whether the practices there are successful in containment in a hospital environment. So I had an image earlier on. I showed the kind of the genome sort of stretched out, an infographic of that. But the virus interacts with our cells on a 3D basis. So virus infects the cell, it replicates, and it produces progeny virus. Okay, so it replicates and it produces more copies of itself. And naturally, what will happen, you will just get some of those progeny viruses. They will just have a mutation, and that's a random process, right? They might have one mutation or two mutations, and it happens all the time. It's a totally normal part of biology. Now, our bodies apply what we call a selection pressure, right? So our bodies are very different to a bat, right? Or structure is very different, or temperature is very different, or immune system is very different. So what happens? The virus infects, it produces all these progeny, these offspring viruses. And the viruses that are better at infecting us, that are better at transmitting between us, they're going to be more successful. So you get a selection event, right? The viruses that are more successful in humans, the more they're going to outcompete their offspring, their brothers and sisters, their brethren, for want of a better term. They're going to outcompete those, and we're going to see more and more of those variants appear. And the more infections there are, then the greater the chance of a more successful variant arising. And this is normal. It's not what we want to see, obviously, but it's normal, and it's evolution in action. It's a normal biological process. And it's why when we're looking at this, we put the grant application into SFI in March of last year, or whenever it was, to look at those variants. So we've done work with the All Ireland Infectious Disease Study, and that's led by Paddy Mallon out of UCD. And we've taken samples from hospitalized patients from hospitals kind of in the Dublin and the southeast region from Wexford. I think it's about 250 samples. And what's really powerful about some of this data is because in the sequencing consortium, normally we keep a real minimal amount of data. We get kind of the age range, gender, where the person lives, date the sample was collected. In this case, within the All Ireland Infectious Disease Study, they have a lot of the metadata, the overarching patient information that they can include with the variant, the sequence variant information. So, you know, the information on outcomes for the patients, or treatments of the patients, or underlying medical conditions for the patients. So this is just a preprint that came out a week or two ago. And this slide is a little bit study, but it's a little bit busy, but it's gives you a good idea of what was in the study overall. So if we get a pointer up here again. So if we look at this axis here, these are just different types of variants. So B.11 is a type of variant. You don't need to focus on that particularly. Along this axis here, we have time. So March of 2020 to about December of 2020. And if you remember, we had the first wave of the pandemic was in about April, May of last year. And the second wave was kind of October, November territory. And in between, during the summer, we had a period where there was relatively few cases in the community and in the hospitals as a result. So the key point here is that over this second wave, we're seeing variants appear on this side that weren't over there. So these circles, the bubbles, they're just an indication of how many of those isolates, how many of those variants that we detected and sequenced out of this sample set. And you'll see some of these variants are in the second wave and not in the first wave. And what that's telling us is that because those variants were in the second wave and not in the first wave, that difference is there. It suggests that the second wave of the infections resulting in hospitalizations. They were largely seeded or caused by multiple travel-related events from countries outside the island of Ireland. It's worth remembering that these are the hospitalized cases. So if you want further information on the project itself, if you go to the Teagasc website and you search for the sequencing consortium there, you'll get to a website and that will provide more information about the nature of the project and some of the documentation linked to it. Fundamentally, by summary of what I was talking about there, we take surplus material that's being collected for the diagnostic testing and we use that for the DNA sequencing. The DNA sequencing itself is through nanopore sequencing for the data generation, and the analysis we piece together those small little pieces to generate the sequence profile for the whole sample. And new variants introduced into Ireland after the summer likely seeded the subsequent infections, and they were detected in the hospital patients. I'm just going to finish on a story that I personally hadn't heard about, and it might be old hat to many of you listening. There's a lot of talk now about how quickly the vaccines developed, how fast the scientific community was to respond. And to me, it kind of highlights the importance of fundamental science, right? Of I call it basic research rather than translational research. So by way of example, when the virus was detected in China, really that was facilitated by genome sequencing. So 10 years ago, this just wouldn't have been possible. What scientists there were able to do, where they were able to take the patient samples, they were able to sequence that, examine it, and find out that they could find this novel virus there. And then they shared that data with the world. And when they shared that data with the world, I mean, what I've heard is that some of the vaccine companies, they had their vaccine designed within, 48 hours, 72 hours. They already knew what their vaccine design was going to look like, and they were able to push into manufacture and clinical trials. This particular story was something I hadn't heard. It's a really good article in the Atlantic by Sam Keane, I saw a couple of months back, and it talked about kind of the first vaccination drive. Back in the late 1700s, early 1800s, people who caught smallpox, it was rife around the globe, and it was a hugely serious disease, killing hundreds of thousands. But what they realized, what Jenner realized, along with others, was that if people were infected with a related disease called cowpox, they never came down with the deadlier smallpox. And the way that they used to do this was they used to take a sore from a patient with cowpox, and they kind of lance that, and then they cut that into the hand, the arm of the recipient, and that person was infected with the cowpox, and they developed immunity against the illness, against smallpox. So one of the technical difficulties at the time, of course, back in the early 1800s, was that there was no cold storage, there was no way of manufacturing the virus. And Spain, they were worried about their colonies in South America, where the virus was rampant, smallpox was rampant. So they came up with a scheme where they took 22 orphans, they took 22 boys, and they put them on a sailboat from Spain across to Venezuela. And they took 2 of those boys and they inoculated them with cowpox. And then 9 to 10 days later, they knew those boys would be, they'd be kind of coming to the end of their infection. So they would take the cowpox from the pore, from the sore, and then they'd infect the next 2 boys. Then 9 or 10 days later, they'd take the cowpox from those 2 boys and pass it on to the next 2 boys. And they did that. And when they reached, I think they left in November and they arrived in Venezuela in March. And when they arrived, the story goes that there was 1 boy who was still infected, and he had a single sore that was still on his body. And they were able to take the cowpox from that and proceed to inoculate, immunize the local population. And that was so successful. They worked through South America, they worked through Asia, Philippines, and into China. It's estimated that they, vaccinated hundreds of thousands of people, and probably saved millions of lives. But it should give us an indication of how far we've traveled in the terms of our fundamental understanding of science. And it should also be a case that the next time somebody complains to you about, getting a small needle prick in their arm for the purpose of the vaccine, or draw any strange anti-vaccination stories about the power of the vaccine, maybe you can correct them and tell them that we've moved quite a bit. And that's it really for me. Just to say thank you to everybody who's involved in the project. They've been, enormously engaged all the time. And just to come back to something Brendan mentioned earlier on, even the manufacturers, the suppliers who were selling reagents for the work, they gave significant discounts for all the reagents. I mean, everybody weighed in on this. There was a real unity of purpose, and it was really lovely to see. So that's it from me, and I think it's back to Roisin maybe for the question.
So thanks so much, John. That was so interesting to see how the different variants have spread across the country as well as just the world. and to show the teamwork that has gone on between scientists all across the country to get such a great resource up and running for us. So if anybody has any questions, you can pop them into the chat. I suppose I have one that I was pondering throughout the talk there. So John, did you find that you sequenced any new variants before the National Virus Reference Laboratory did? Like, did you have proof that variants were here in the country before we thought they were?
No, so Killian's guys had, Killian Gascon's team, Michael Carr and Gabo there, they would have had first access to those community samples. So they would have detected those initially. I mean, the screening anyway, the initial screening is all done off the PCR test anyway. And we didn't, it's not like we spontaneously developed the virus in this country, right? The virus that we were seeing, the variants we were seeing were being introduced from outside the island of Ireland.
Yeah, so another question there is that it was mentioned in the talk on Monday night that we had that, that the between 5 and 10% of samples should be sequenced. So I was just wondering, do you think it's something that would be feasible to run in the clinical laboratory to have medical scientists sequencing the different variants that come in?
Yeah, I think it's something that is doable. I think the challenge around something that arises in the manner like this is that you have to have some of the infrastructure in place already. Okay, so you need to develop the method for doing the sequencing to target whatever it is you're sequencing. But you also have to have the instrumentation in place. You have to have the ancillary equipment for running a sequencing lab. You have to have the experience of doing it. You have to have the informatics. You have to have the compute in place. The UK has done a fantastic job of sequencing the variants, but that's because they've had hundreds of millions of pounds worth of investment over the last decade. So they were ready to go, and they've got a really strong and powerful genomics community in the UK. So it's certainly doable, but you have to have that infrastructure set up and the skills set up. It's not something you can kind of do in a clinical setting straight away.
Brendan, do you have any thoughts on that as a former medical scientist yourself?
Yeah, this was when I started in medical science, molecular techniques were not part of the routine lab, and they now very much are. And even to the point of not just closed box systems and CE marked closed systems, but open, finding primers and setting up assays. Certainly you wouldn't want to underestimate, because I have a very remote understanding of sequencing, and it is very data heavy. It's very you need the support behind that, the analytics behind that, but it is of huge value. And I think ultimately we'll either need to set up a national repository or service for sequencing outbreaks, or manage them locally. But it is the key, when we start getting all single occupancy rooms for patients, the next natural step is to trace the journey of organisms across an organization and understand where they're coming from, how they're mutating, and what the new challenges are. So I don't think we're there yet. But we're starting to get there. I think the human genomic side will come first because of the cancer diagnostics and that, and the value of drugs will support that as well. The cost of drugs and wanting to make the best use of those, best outcomes for patients.
Great. Another question there is, could you describe the ethical approval and consent process that you had to go through for sequencing? Do the results go back to the patients? So, for example, do they know that they have that particular variant?
Yeah, so that's a really good question, right? So the single biggest delay we had with the project had to do with the bureaucracy, and that's something Brendan mentioned earlier on. So for the consortium itself, we keep a very minimal amount of data, right? We'd regard it as pseudonymized data. So that means that in theory, because the hospital still holds the patient information, that the variant data could be linked back to the particular patient, but we never get that data. So we had to go through ethical approval from the National Research Ethics Committee to be able to do the sequencing side of things. And we were very specific about the data we were going to collect and make public so that it couldn't be linked back to a particular patient. It is conceivable that somebody could contact the hospital and see if their particular patient information, if their particular sample was sequenced. And because the hospitals hold the information on that sample, they could get us to provide information on which the variant, which sample it was, and then they could provide that back to the patient. But it's all kept at an arm's length, which means that, you know, any of the scientists working in the lab, we can't know where the sample came from originally, which patient it came from.
That's very interesting to think you'd be able to track back your sample to see where it might have come from. So just, is there any more questions to come in? If not, we be ready to finish up, but I wouldn't want anybody to miss out on getting their question answered. No, so we'll leave it there. A big thank you to everyone for coming, and especially to John and Brendan for speaking, and to the societies and everybody who helped us organise it. And I hope you enjoyed it and have a lovely Friday evening and the rest of your weekend. So thank you, everyone.
Thank
Well
you very
done,
much.
thank you.
Thank you so much.